Welcome to the Creative Commons RX podcast. Before diving into this episode, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.
Welcome back to The Deep Dive. We're the place where we try to crack open these really complex subjects, give you the core framework you need to, get your head around them.
That's the goal.
And today we are diving deep into what feels like a truly revolutionary frontier: cellular and genetic therapy, CGT.
Yeah, it really is transformative stuff.
We're not just talking about managing chronic illness anymore, are we? This is, well, it's a whole new class of therapeutics, potential single-dose cures maybe, changing the game for diseases from like rare single-gene disorders right through to complex cancers.
It's arguably the biggest shift we've seen in pharmacology since maybe monoclonal antibodies. But, and this is the big but, it's an incredibly intricate field, really complex.
Okay.
So for you listening, if you want to understand the investment side, the risk, the clinical potential, you really need to get a handle on the foundational principles, the jargon, the key terms, and these strategic distinctions that really define the landscape.
Okay, let's unpack this then. Our goal today is precision, really. We want to nail down that core framework for you. Look at the major tools, the vectors, what they mean for biological risk, and also those hidden logistical hurdles manufacturing monitoring that make these treatments so complex and often very expensive.
Right. It's about providing that structural knowledge so you can differentiate between the players, the approaches in this whole new world of genetic medicine. Okay, so to start, maybe we should really clearly define the two foundational bits that are, driving all this therapeutic development right now.
Sounds good. Let's start with cell therapy. When we say that, what's the actual active therapeutic agent? What is the drug?
The drug is the living cell itself. That's the core idea. The goal is administering these cells, and they can be from the patient, that's autologous, or from a donor, allogeneic, to repair something, replace something, or augment a function. The cell does the work.
And it's interesting because cell therapy, it's not brand new, is it? This basic concept goes back decades.
Oh, absolutely. Decades. The concept is, well, ancient if you think about it, but the techniques are always being refined. A simple blood transfusion, that's cell therapy.
Right.
More complex and really foundational to this whole field is hematopoietic stem cell transplantation. The classic bone marrow transplant.
Sure. Standard for a long time.
Exactly. Standard practice for decades for certain blood cancers. You're replacing a faulty blood system. Cells are harvested, maybe stored, reinfused. But critically, there's no genetic modification involved there. It's the cell's natural ability we're relying on.
You mentioned Prevenge earlier. That's an approved therapy for advanced prostate cancer. Is that a good example of a modern cell therapy without the genetic engineering piece?
Perfect example. Prevenge involves taking the patient's own immune cells, processing them outside the body, activating them against a tumor antigen, and then putting them back. The cell is trained, you could say, but no new genetic code is permanently added.
Okay. So that's cell therapy. Now let's contrast that with gene therapy. If the cell is the drug in the first case, what's the drug here?
Here, it's purely a molecular intervention. The therapeutic agent is the genetic material itself. DNA or RNA delivered like a biologic drug.
Okay.
The whole point is to fix the problem at its source, right? To modify or manipulate gene expression. Maybe replace a faulty gene, maybe switch off one that's causing trouble, or introduce a completely new gene to provide some benefit. It's about correcting the blueprint.
Right, going right to the genetic code itself, which then sets up the complexity, doesn't it? The convergence point. Gene-modified cell therapy. This is where things get really interesting and maybe really complicated.
This is the hybrid.
Yeah.
And it's essentially synonymous with what we call ex vivo gene therapy modification outside the body.
Ex vivo, got it.
We're leveraging the cell's natural abilities to survive, travel, and multiply, but we first give it new genetic instructions. You could think of the cell as the delivery truck, maybe.
Okay.
And the gene is the cargo, the payload. That tells the truck what job to do when it gets where it's going.
And the classic example here, the one everyone points to, is CAR T-cell therapy, Kymriah, Yescarta. These therapies that have shown, frankly, amazing results in some leukemias and lymphomas. How does CAR T blend both cell and gene therapy?
It's a really great illustration because it's multi-stage. First, you collect the patient's T cells. That's done via a process called a pheresis. That's your cell therapy component.
Right, harvesting the cell.
Exactly. Second, those T cells go off to a specialized manufacturing lab. There, they get engineered. Usually, a viral vector is used to introduce a new gene that codes for something called a chimeric antigen receptor, or CRR. That's the gene therapy component, adding the new genetic instruction.
And that CRR is what lets the T cell recognize the cancer.
Precisely. That new gene permanently equips the T cell. The result is this modified T cell, this living drug. It can now recognize and ideally eliminate cancer cells that have a specific marker on their surface, usually CD19 for those therapies.
And it's this whole process, harvest, engineer, expand, reinfuse, that drives the complexity.
Absolutely. That personalized manufacturing, that need to handle living cells for each individual patient, that's what makes it logistically incredibly challenging and contributes significantly to the high cost. It's a world away from popping a pill.
Okay, so we've figured out what we want to deliver a cell, a gene, or this gene-modified cell. The next big strategic question is, where do we do the modification? Inside the body or outside? And that leads us to this critical split in vivo versus ex vivo. What's the main factor driving that choice?
It really comes down to biology, purely biological determinants. The accessibility of the target tissue and its regenerative potential. Can we actually get the cells out easily and put them back safely?
Right.
So the ex vivo strategy, modifying outside the body, is really only feasible if we can safely remove the target cells, modify them, grow them up, expand them, and then reintroduce them without causing major harm.
Which practically limits it to certain cell types.
Pretty much. It largely restricts ex vivo approaches to the hematopoietic system, blood stem cells in the immune system, like T cells, things we can get relatively easily from blood or bone marrow.
Okay, so if the target tissue is, say, internal, solid, maybe doesn't regenerate easily, like the brain or the heart or muscle or the eye, then we're basically forced into the in vivo approach, meaning within the living.
That's really the only option then, yeah.
Yeah.
The core idea of in vivo gene therapy is direct delivery. You put the vector carrying the therapeutic gene straight into the patient. The target cells get modified right there in their natural spot within the body.
And how is that actually done? How do you administer it to make sure it goes to the right place? Precision seems key.
It is absolutely key. Delivery can be systemic, like an IV infusion. If we use a vector, say an AAV vector, that naturally likes to go to the liver, we can sometimes just infuse it systemically.
Okay.
The vector finds its way to the liver cells, delivers the gene, and maybe starts producing a missing protein, like a clotting factor for hemophilia. That's the hope for therapies like Roctavian or Hemgenix.
But for other organs, like the eye or the brain, systemic doesn't seem like it would work well.
No, it's often inefficient and could be quite risky. Too much off-target delivery. That's why you see therapies like Luxturna for inherited blindness. That requires a very precise, highly localized injection under the retina, subretinal, to make sure only those target retinal cells get the AAV vector.
And Zolgensma for spinal muscular atrophy. That's systemic, right? But targeted.
Right. It uses a specific type of AAV, AAD9, which is known to be better at crossing the blood-brain barrier, especially in infants. So it's given as a systemic IV infusion, but the vector serotype itself helps target it towards the motor neurons that need the functional SMN1 gene.
Logistically for the patient, this in vivo approach sounds simpler, doesn't it? Often just a single administration, maybe an infusion or an injection. But what's the downside? What's the trade-off, especially thinking about the immune system?
The big trade-off is immunogenicity risk. It's a major one. When you introduce a viral vector, especially systemically, the patient's immune system often sees that viral capsid, the protein shell of the vector, as foreign.
Makes sense.
And it mounts an immune response. It produces neutralizing antibodies, or NAbs.
And why are these NAbs such a problem, particularly for in vivo therapy staying effective over time?
Well, two main reasons. First, the immune system might just wipe out the vector before it even gets to the target cells properly. That limits how effective the therapy is right off the bat.
Okay.
Second, and this is really crucial for the long term, Once a patient develops high levels of these NAbs against a specific AAV serotype, they are essentially locked out from ever being treated again with that same type of AAV.
Wow, okay, so no redosing.
Generally, no. If the gene expression fades years later, or maybe a better therapy comes along using the same AAV type, they can't get it. We even have to screen patients beforehand. If they already have high levels of NAbs from a past natural infection, which is quite common in the population, they might not be eligible for the therapy at all.
That really limits the potential patient pool and future options. Okay, that's a serious constraint. Now, let's pivot to ex vivo, outside the living. If in vivo is more like a single shot, ex vivo sounds like a much more involved, multi-step, highly controlled process, almost like surgical reengineering.
It is. It requires a huge amount of coordination and planning. We're dealing with those accessible cells, typically hematopoietic stem cells, HSCs, or T cells. The whole process is indirect.
Yeah.
You harvest the cells, ship them off, modify them, grow them, test them, ship them back, prepare the patient, and then reinfuse.
Walk us through the patient side of that. The burden sounds significant. It seems like it could take months.
It often does. First, the patient's cells are collected. For HSCs, or T cells, that's usually pheresis, filtered in the blood. Then those cells are shipped, often cryopreserved, maybe across continents, to a specialized centralized manufacturing facility.
Okay.
This is the centralized manufacturing phase. Modifying the cells with the vector, expanding them potentially into billions of cells, doing quality checks, and then freezing the final product. That whole manufacturing process can take anywhere from, say, 4 weeks to maybe 12 weeks or even more.
And what's the patient doing during those weeks or months while their cells are away being modified?
They're waiting, and then preparing for what is often the toughest part, conditioning. For HSC therapies especially, this usually means hospitalization for high-dose chemotherapy.
To make space for the new cells.
Exactly. You have to destroy the patient's existing bone marrow, essentially, to create a niche for the newly engineered stem cells to engraft and take hold. Only after the patient gets through that conditioning and recovers a bit from the acute toxicity can the modified cells finally be thawed and reinfused.
That conditioning step alone.
Oh,
That sounds like where a lot of the immediate risk and cost comes from. Major morbidity.
it's often the most acutely dangerous part of the journey for these HSC-based therapies, yes. The goal of that myeloablative chemotherapy is profound immunosuppression, wiping the slate clean.
Right.
The risks are substantial during that period. Extreme vulnerability to potentially deadly infections, severe mucositis, which is incredibly painful inflammation throughout the digestive tract, needing lots of transfusions for platelets and red cells. The patient can be in the hospital, often in isolation, for weeks just recovering from the conditioning before the new cells even start to work. Okay, so that choice we talked about in vivo versus ex vivo modification, it pretty much dictates the kind of delivery vehicle, the vector we have to use. And that choice, the vector, has huge implications for how long the effect lasts, and crucially for the safety risks involved.
And viral vectors are still the main workhorses, right? How do scientists actually take something that evolved to cause disease and turn it into a safe therapeutic tool?
It's sophisticated molecular engineering, really. Viruses are naturally brilliant at getting genetic material into our cells. So we take the virus and we essentially gut it. We remove the genes that allow it to replicate and cause disease.
Okay.
Then, in that empty space, we insert the therapeutic gene we want to deliver the gene cassette. So the vector keeps the machinery to get into the cell, but it can't make more copies of itself or make you sick. It becomes just a delivery shell, a very efficient one.
Let's start with the big one for in vivo delivery, AAV, adeno-associated virus. We know it works well in cells that don't divide much. What actually happens to the AAV's genetic payload once it gets inside the cell nucleus?
AAV is interesting. It's small, it's stable, it delivers a single strand of DNA. Once that gets into the nucleus, the cell's own machinery converts it into a double-stranded DNA molecule. And then this molecule forms a stable, closed loop it circularizes. We call this structure an episome.
An episome. And why is that episomal state so important from a safety point of view?
Because it typically just sits there in the nucleus, separate from the host cell's chromosomes. It doesn't insert itself into the patient's own DNA.
Ah.
And that means AAV carries a very low risk of causing insertional mutagenesis, which is the danger of disrupting an important host gene by inserting foreign DNA. That's its main safety advantage.
And that helps with durability, too.
In the right cells, yes. Because cells like neurons or muscle cells or those retinal cells, they don't divide often. or at all. So that stable episome can just hang out in the nucleus for a very long time, potentially for years, maybe even decades, allowing continuous production of the therapeutic protein from that single initial dose.
We mentioned AAV serotypes before, different flavors of AAV. Why is having that diversity so critical for targeting specific tissues?
The serotype refers to the slightly different protein coat, the capsid, on the outside of the AAV. And each serotype has a different natural preference, or tissue tropism.
Like a postcode system.
Sort of, yeah. For example, AAV2 seems to work well for the retina, which is why it's used in Luxturna. AAV8, or maybe AVH10, might be better for targeting the liver. AAV9, as we said, is good for crossing the blood-brain barrier into the CNS. So being able to swap out the capsid shell while keeping the same therapeutic gene inside gives us really valuable control over where the therapy primarily goes in the body.
But AAV isn't perfect. What are its main sort of unavoidable limitations?
Two big ones. First, size matters. AAV has a pretty small packaging capacity. Only about 4.7 kilobases kb of genetic material can fit inside.
Okay.
So if the gene you need to deliver is really large, the classic example is the full-length dystrophin gene for Duchenne muscular dystrophy, which is huge, around 14 kilopounds. Well, it just won't fit into a standard AAV. That's a hard physical limit.
Right.
The second major limitation, as we discussed, is that immunogenicity. The pre-existing antibodies in many people and the inability to redose effectively once antibodies develop, that's a big hurdle.
Okay. Now let's switch gears to lentiviral vectors, LVs. These are the backbone for many ex vivo therapies like RRT, often derived from HIV, but heavily engineered for safety. Their defining characteristic is that they do integrate into the host genome. Why take that riskier approach? Why is integration necessary?
Integration is absolutely essential if you're targeting cell populations that divide actively and continuously, like hematopoietic stem cells or T cells.
Why is that?
Think about it. If you modify a stem cell and that stem cell is going to divide potentially thousands or millions of times to create all the different blood cells, you need the therapeutic gene to be copied along with the cell's own DNA every single time it divides.
Okay. Otherwise it gets diluted out.
Exactly. If the therapeutic gene was just sitting there as an episome, like with AAV, it wouldn't be reliably passed down to all the daughter cells. It would get lost over time as the cell population expands. Permanent integration ensures the correction is stable and inherited by all the progeny cells, hopefully for the lifetime of the patient.
But that integration brings back the big safety concern, insertional mutagenesis. You mentioned this risk is real. How does it happen, and how does doing the modification ex vivo help manage that risk?
The LV integration isn't perfectly targeted. It's described as semi-random. So there's always a small but real risk that the vector might happen to insert itself near a gene that controls cell growth, maybe activating a cancer-promoting gene, a proto-oncogene, or shutting down a gene that normally prevents cancer, a tumor suppressor gene.
And this has actually happened.
Tragically, yes. In some early gene therapy trials, particularly for X-linked severe combined immunodeficiency, XSCID, using related integrating vectors, some patients develop leukemia years later as a direct result of insertional mutagenesis.
So how does ex vivo help?
The ex vivo setting gives us a critical control point. We modify millions or billions of cells in the lab before they go back into the patient. This allows for extensive quality control and safety testing on the final cell product. We can analyze the integration patterns in the pool of cells, check if the cells are growing normally in culture, look for any red flags suggesting a dangerous integration event might have occurred in a particular clone.
I see. You can screen the batch before infusion.
Precisely. That kind of detailed pre-infusion safety assessment is simply impossible when you inject the vector directly into the patient in vivo. The ex vivo process allows for that crucial safety check.
Okay, moving beyond viruses for a moment. There are also non-viral systems being developed, often aiming for maybe better safety or easier manufacturing, but maybe with different durability. Let's touch on mRNA delivered via lipid nanoparticles, LNPs. We all became familiar with this thanks to the COVID vaccines.
Right. LNPs are basically tiny fat bubbles, synthetic shells, designed to protect the very fragile mRNA molecule and help get inside the cell. The defining features here are really speed and safety.
How so?
Once the LNP fuses with the cell membrane and releases the mRNA into the cytoplasm, the main body of the cell's own ribosomes immediately grab it and start translating it into the desired protein. Critically, the mRNA never enters the cell nucleus.
So, zero risk of messing with the patient's DNA.
Exactly. No interaction with the genome means no risk of insertional mutagenesis, no genotoxicity. That's a huge safety advantage. Plus, mRNA is naturally unstable. The cell breaks it down within hours or maybe a few days.
Right.
This makes LNP mRNA delivery incredibly safe from a genetic standpoint and very fast and scalable to manufacture, as we saw during the pandemic. But that lack of durability is also its limitation, right? It's great for vaccines or maybe transient therapies, but not suitable if you need lifelong protein production after a single shot. For chronic conditions, you'd need repeated doses, potentially forever.
Okay. And the last tool in this part of the kit, antisense oligonucleotides, ASOs. They aren't delivering a whole gene, are they? More like regulators.
That's a good way to put it. ASOs are short, synthetic, single strands of nucleic acid like DNA or RNA, but engineered. They are designed with incredible specificity to bind only to one particular target RNA molecule inside the cell, usually either a messenger RNA, mRNA, or the precursor pre-mRNA.
And what happens when they bind?
Depending on how they're designed, they can do a couple of things. They might trigger an enzyme called RNase H to come and chew up the target mRNA, effectively silencing the production of the protein encoded by that mRNA. Or they can bind to pre-mRNA and subtly change how it's spliced, maybe correcting a splicing defect that was causing disease.
Useful for specific types of genetic errors, then.
Very useful, especially for modulating splicing or silencing toxic genes. But like mRNA therapies, the effect of ASOs is transient. They get cleared from the body over weeks or months. So for a chronic condition, treatment typically requires repeated injections, often lifelong.
All right, so we've covered how the delivery vehicles, the vectors, viral and non-viral, and the where in vivo versus ex vivo. Now let's get to the what. What is the actual molecular goal? What are we trying to achieve at the DNA or RNA level inside the patient cells? We can break this down into maybe 3 main strategies or goals.
That sounds right. The first, and perhaps the most intuitive, is gene replacement, or sometimes called gene addition. This is mainly for tackling those loss-of-function genetic disorders, where a gene is missing entirely or it's present, but broken, defective.
So the classic idea of just putting back the missing piece, like providing a correct blueprint.
Exactly. You deliver a healthy functional copy of the gene. We call this the transgene.
Yeah.
It doesn't usually replace the faulty gene. It just coexists alongside it, often as one of those AAV episodes we talked about. The cell then uses this new correct template to produce the functional protein that was missing or deficient. We see this with Zolgensma, providing a working SMN1 gene for SMA patients, or Luxturna, providing a working RPE65 gene for patients with that specific form of inherited blindness. Hemophilia therapies like Roctavian and Hemgenix aim to do this in liver cells, delivering a gene for factor VIII or factor IX.
Just adding the correct copy makes sense. Now, the second goal is kind of the opposite. Gene silencing or gene deletion, often called knockout. Here, the aim is to inactivate a problematic gene.
Right. This is typically for diseases caused by a toxic gain-of-function mutation, where the mutated gene produces a harmful protein, or maybe where a normal protein is just being overproduced and causing problems.
And how do you achieve that silencing or deletion?
Well, you can achieve temporary silencing using tools like ASOs or perhaps Cernoins, which work by degrading the target mRNA. There's research ongoing using ASOs to try and silence the toxic Huntington gene in Huntington's disease, for example.
But that's temporary. What about a permanent switch off, a permanent knockout?
For a permanent effect, you generally need gene editing tools, with CRISPR-Cas9 being the most well-known right now.
And how does CRISPR achieve a knockout? Is it precise deletion?
It's precise in where it cuts, but the knockout itself relies on the cell's slightly messy repair process. The Cas9 enzyme acts like molecular scissors, guided by an RNA molecule to a very specific DNA sequence in the target gene. It then makes a clean cut through both strands of the DNA, a double-strand break, or DSB.
Okay, it cuts the DNA. Then what?
The cell absolutely hates having broken DNA. It flags the DSB as a major emergency and rushes to repair it. The main repair pathway it uses for this kind of break is quick, but notoriously error-prone. It's called non-homologous end joining, or NHEJ.
Error-prone. So it doesn't always fix it perfectly.
It's exactly. NHEJ often makes small mistakes when sticking the broken ends back together. It might insert a few extra DNA letters or delete a few. These small insertions or deletions, indels, often cause a frameshift mutation. This scrambles the genetic code downstream from the cut, essentially garbling the instructions so the cell can no longer make a functional protein from that gene. Voila, you've achieved a permanent knockout.
And the therapy Casgevy for sickle cell disease and beta thalassemia uses this knockout strategy, but in a really clever, indirect way, right? It doesn't fix the sickle gene itself.
That's the brilliance of it.
Yeah.
Sickle cell is caused by mutation in the adult hemoglobin gene. But we all have another hemoglobin gene, the fetal hemoglobin gene, which works perfectly fine and doesn't sickle. Normally, that fetal hemoglobin gene gets switched off shortly after birth by a repressor protein. This repressor is encoded by a gene called BCL11A.
Okay.
So what Casgevy does, using CRISPR-Cas9 ex vivo in the patient's own hematopoietic stem cells, is it knocks out the BCL11A gene. It cuts and inactivates the gene that makes the repressor.
So by removing the off switch,
You effectively turn the fetal hemoglobin gene back on. The stem cells start producing high levels of fetal hemoglobin again, which compensates for the faulty adult hemoglobin and essentially cures the clinical symptoms of sickle cell disease or beta thalassemia. It's a knockout strategy, but targeting a regulatory gene, not the disease gene itself. Very clever.
very clever indeed. Okay, that brings us to the third and perhaps most technically challenging goal. Gene addition, or insertion, sometimes called knock-in. This sounds like real gene surgery, precisely putting something new into a specific spot. Why is this so much harder than just knocking a gene out?
It's much harder because a knockout relies on that efficient but error-prone NHEJ repair pathway. A knock-in requires something much more sophisticated from the cell.
Yeah.
You still use CRISPR to make the precise DSB at the desired location, but then you also have to provide the cell with a donor DNA template, basically, the piece of DNA you want to insert.
Okay.
And you need the cell to use that template to repair the break, accurately inserting the new sequence. This forces the cell to use a different, much more precise repair pathway called homology-directed repair, or HDR.
And HDR is less efficient.
Much less efficient than NHEJ, unfortunately. HDR really only works well when the cell is actively dividing, and it requires that the donor template has arm sequences on either side of the inserted piece that perfectly match the DNA sequences flanking the cut site in the genome. Getting the cell to preferentially use your template and the HDR pathway instead of just quickly patching the break with NHEJ is the major technical hurdle. It happens, but often at low frequencies.
But if you could master HDR and achieve efficient knock-in, that would open up amazing possibilities for control, wouldn't it?
Absolutely. That's the holy grail for precision. If we could reliably use HDR, we could, for example, insert a therapeutic gene, like the CAR gene for CAR T therapy, directly into a specific known safe harbor location in the genome. Maybe into a spot like the TRAC locus in T cells, which naturally gets switched off when the T cell receptor is made.
And what would be the advantage of that?
You get much more uniform, predictable, and potentially safer expression of the CRR compared to the somewhat random integration you get with lentiviruses. You'd know exactly where the gene went, and you could potentially control its expression levels better. It really represents the next level of genomic precision we're aiming for.
Okay, the science is incredible, the potential is clearly there. But as we've touched on, the logistical hurdles are just enormous, especially for those ex vivo therapies. Let's circle back to the patient journey for a moment, specifically that conditioning step needed for the HSC therapies. You mentioned myeloablative chemotherapy wiping out the marrow. What does that actually mean for the patient in terms of toxicity?
Yeah, it's really important not to understate this. It's not a trivial procedure. It's a profound medical intervention with significant risks. The high-dose chemo is designed to completely eliminate the patient's existing bone marrow. This leads to a period where they have virtually no blood cells, severe pancytopenia, no infection-fighting white cells, no clot-forming platelets, no oxygen-carrying red cells.
So they're incredibly vulnerable during that time.
Extremely vulnerable. This necessitates weeks of hospitalization, often in strict protective isolation rooms, to minimize infection risk because they essentially have no functioning immune system.
And the risks aren't just infection.
No. We're talking about potentially life-threatening bacterial or fungal infections that can rapidly escalate to sepsis. But there's also the direct, often brutal toxicity of the chemo itself. Severe nausea, vomiting, hair loss, debilitating fatigue, and that awful mucositis, the painful inflammation and ulceration of the entire lining of the mouth and gut. Plus, they are completely dependent on blood and platelet transfusions for weeks until the new stem cells hopefully engraft and start working. That conditioning phase is often the most dangerous, most difficult part of the whole process for the patient.
Wow. Okay. Now, for CAR T therapies, the conditioning is usually a bit less intense, right? But the focus shifts to managing unique toxicities after the CAR T cells are infused. You hear about cytokine release syndrome, CRS, and ICANS. What are those? What do they actually look like?
Right. These are specific, potentially life-threatening side effects related to the massive activation of the engineered immune cells. CRS is basically a runaway systemic inflammatory response. The activated CRT cells release a flood of signaling molecules called cytokines.
Okay.
This can range from mild flu-like symptoms, fever, chills, fatigue, to a severe critical illness that looks a lot like septic shock. We're talking high fevers, dangerously low blood pressure, leaky blood vessels, difficulty breathing, organ dysfunction. It requires immediate recognition and often aggressive treatment, typically with drugs that block a specific cytokine called IL-6, like tocilizumab.
And ICANS, that sounds like it affects the brain.
It does. Immune effector cell-associated neurotoxicity syndrome. It's thought to be related to the inflammatory cytokines and maybe the CRT cells themselves, affecting the central nervous system. Clinically, patients can present with a whole range of neurological symptoms: confusion, disorientation, difficulty speaking, aphasia, tremors, seizures, and in severe cases, even swelling of the brain, cerebral edema.
Also potentially life-threatening then.
Absolutely. These are unique, sometimes unpredictable toxicities that really require specialized hospital teams, often including neurologists, and very intensive monitoring, particularly in the first couple of weeks after infusion.
Okay, let's shift from the patient bedside to the manufacturing facility. You mentioned this batch of one problem for autologous therapies, where every patient is their own manufacturing run. This must make chemistry, manufacturing, and controls, CMC, incredibly complex.
It's a regulatory and logistical nightmare, frankly. It leads to this paradigm where regulators say the process is the product. Because you're dealing with living cells and every patient's starting material is slightly different, even tiny variations in the manufacturing process, a different batch of reagent, a slight change in temperature, how long cells sit in a centrifuge could potentially alter the quality, safety, or effectiveness of the final cell product.
So consistency is extremely difficult to achieve.
Extremely. This is why scaling up autologous therapies is so challenging and expensive. The entire supply chain, from collecting the cells, shipping them, often internationally under strict cold chain conditions, manufacturing, testing, and shipping back. It requires meticulous tracking, documentation, and coordination between many different partners.
When that final product is ready, before it goes back to the patient, what are the key things regulators look for? What are the most critical quality attributes, CQAs?
There are several, but maybe three stand out. First is purity, making sure the product contains what it's supposed to contain and minimal contaminants. An absolutely crucial part of purity testing for viral vector products is checking for replication-competent virus, RCV.
Meaning the vector hasn't somehow regained the ability to copy itself.
Exactly. If the engineered, supposedly non-replicating vector accidentally reverted back to a form that could replicate inside the patient, that could potentially cause a widespread, dangerous infection. RCV testing is mandatory and rigorous.
Okay, purity. What else?
Second is viability. Pretty straightforward. The cells have to be alive and functional after being thawed. The FDA generally sets a threshold, often recommending at least 70% viable cells for infusion. Dead cells aren't going to help.
Makes sense.
Third, and this is often the most difficult CQA to measure reliably, is potency. Potency isn't just about cell count or purity. It's a measure of the product's actual biological activity. Does it do what it's supposed to do?
How do you measure that?
It usually requires a specific biological assay. For CAR T cells, for example, the potency assay might involve testing the ability of the manufactured CAR T cells to actually kill target cancer cells in a lab dish, in vitro. Developing a potency assay that is robust, reproducible, and truly predictive of how the therapy will work in the patient is a massive challenge in this field.
Immense challenges. Okay, finally, let's talk about the regulatory oversight after the treatment. This idea of long-term follow-up, LTFU. Why are regulators like the FDA mandating patient surveillance that can last 15 years for a treatment that might be given just once?
The core reason is the potential permanence, or at least the very long-lasting nature of these therapies, combined with the fact that we're intervening at the genetic level. While the acute risks, like CRS or conditioning toxicity, are managed in the short term, there's a theoretical risk of delayed adverse events cropping up years or even a decade or more down the line.
Primarily the cancer risk from integration.
That's the main one, yes. Secondary malignancies arising from genotoxicity, like insertional mutagenesis. We simply don't have decades of experience with most of these specific therapies yet to know the true long-term frequency of such events. So the long-term follow-up is essentially a safety surveillance program.
And the duration of that required follow-up isn't the same for all types of gene therapy?
That's right. The regulators have taken a risk-based approach. The most stringent requirement is 15 years of follow-up. This is currently mandated for products considered to have the highest theoretical genotoxic potential. Specifically, therapies using integrating vectors like lentivirus and all gene editing products, including those using CRISPR-Cas9.
Why both integrating vectors and gene editing?
Because both involve permanently altering the host genome. Lentivirus integrates semi-randomly. CRISPR-Cas9, while targeted, creates double-strand breaks, which can sometimes lead to unintended larger genomic rearrangements or off-target cuts elsewhere in the genome. So that extended 15-year surveillance is deemed necessary to monitor for any potential delayed consequences.
And for the non-integrating vectors, like AAV?
For AAV vectors, which generally exist as non-integrating episomes and are considered to have a lower intrinsic risk of altering the host genome in a dangerous way, the typical recommendation is for 5 years of follow-up. It's still substantial, but it reflects that perceived lower long-term risk profile compared to integrating vectors or gene editing.
So the length of follow-up reflects the perceived risk of the technology used.
Precisely. It's the regulatory system trying to balance the incredible therapeutic potential with the need for caution and long-term safety monitoring, given how new these powerful technologies are. And it's crucial to remember we're only talking about somatic gene therapy here. Changes made only to the patient's non-reproductive cells, changes that cannot be inherited. Germline gene therapy, which would alter sperm or eggs and be heritable, is currently prohibited worldwide due to profound ethical and technical concerns. Hashtag outro.
Wow. Okay, this has been a really deep dive, but hopefully it's provided you, the listener, with that essential roadmap we promise for understanding this incredibly complex field of genetic medicine. We've really tried to unpack that core strategic split, haven't we?
Yeah, the dichotomy between, on one hand, AAV vectors used in vivo, often for gene replacement in more static, non-dividing tissues like the liver, eye, muscle, maybe brain,
Right.
versus, on the other hand, the lentiviral vectors and gene editing tools primarily used ex vivo to really re-engineer the highly dynamic hematopoietic and immune systems, the blood and immune cells.
It's truly transformative science, no question. But it's clearly matched, step for step, by just immense logistical and manufacturing complexity.
Absolutely. And that's really where the next wave of innovation is focused, trying to overcome the limitations and risks we've discussed today. We're seeing really exciting progress in advanced gene editing technologies, for instance, moving beyond CRISPR-Cas9, which relies on making that double-strand break.
Which carries some risks.
Exactly. Newer tools like base editing and prime editing aim to make precise changes, like correcting a single DNA letter or inserting a small sequence without creating that potentially problematic double-strand break in the first place. The hope is that these next-generation editors will be inherently safer, with fewer off-target effects or unwanted genomic rearrangements.
Making the whole process more precise and potentially less risky. And what about the batch of one problem, making these therapies more accessible?
That's the other huge frontier, scalability and accessibility. That personalized autologous model is just incredibly expensive and logistically challenging. The ultimate goal for many companies and researchers is to develop allogeneic, off-the-shelf cell therapies.
Using donor cells.
Right. Using cells from healthy screened donors to manufacture large, standardized batches of therapeutic cells that could potentially treat many patients. The big challenge there, of course, is figuring out how to prevent the patient's immune system from rejecting these foreign off-the-shelf cells or preventing the donor cells from attacking the patient, graft versus host disease. If those immunological hurdles can be overcome safely, it can dramatically lower costs, eliminate the long waiting times for manufacturing, and make these therapies available to far more people globally.
A huge goal. Now, we ended the last section talking about that long-term follow-up up to 15 years. It strikes me that the patient receives this potentially curative therapy, often in a single infusion or procedure.
Yeah.
And then they are essentially signing up for a decade and a half of monitoring and surveillance.
That commitment is really the long shadow of risk that defines where we are right now with genomic medicine, especially for the integrating and gene-edited therapies. Our complete understanding of the long-term safety profile relies entirely on patients participating faithfully in these extended follow-up studies, coming back for checks year after year.
Which must be a challenge.
It's a profound ethical and logistical challenge. How do you maintain that engagement and ensure effective surveillance over 15 years for someone who might feel perfectly healthy and cured just months after treatment? They receive their miracle, but the scientific and regulatory process requires this long tail of observation. Managing that long-term patient commitment is, in many ways, the unseen but absolutely essential component underpinning the responsible development of these potentially transformative cures.
A really critical point to end on as this field continues to evolve at such speed. Well, thank you for guiding us through that incredibly complex but crucial framework today.
My pleasure.
And for you listening, hopefully you now feel equipped with the core concepts you need to really understand and navigate this new era of genetic medicine. Thanks for joining us for the deep dive.
This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.